So let's start today's discussion. This discussion will be on the properties of the nucleic acids. Alright, so when I say nucleic acids, what do I mean? What are nucleic acids? So nucleic acids will be the DNA and the RNA. Know this? The nucleic acids will be DNA and the RNA. So we'll take them one by one and discuss the DNA and the RNA. Obviously, let's start the DNA first, and then we'll talk about the properties of the RNA. When we talk about the eukaryotic cell in the humans, when we talk about the DNA, we find that this DNA is present in different compartments. Based on that, we give the names in which compartment they are present. Alright, so talking about the DNA in the eukaryotes, rather in humans, the DNA is found in the nucleus. Majority of the DNA will be found inside the nucleus of the cell. Alright, more than 99.9%. More than 99.9% of the cellular DNA will be in the nucleus. Alright. In addition to this, obviously you know that we have a small amount of DNA in the mitochondria. We have a small amount of DNA inside the mitochondria. This content is less than 0.1%. This content is less than 0.1%, and together the nuclear and mitochondrial DNA make up the cellular DNA. Alright, so within the cell, the DNA will be present either in the nucleus or in the mitochondria. Majority of the DNA is obviously present inside the nucleus. However, recently there are certain techniques which have been developed which do not make use of either the nuclear DNA or the mitochondrial DNA. So now we have a third category of DNA which we have to know about. However, please note that DNA is not present inside the cell. When we are detecting it, we are measuring it, it is not present inside the cell. Although it is coming from the cell, it is not present inside the cell. We call it the cell-free DNA. We call it the cell-free DNA, sometimes written as cfDNA, the cell-free DNA. The new techniques have been evolved making use of the cell-free DNA. So what I'll do very briefly, I'll mention about the cell-free DNA and then we'll go back to the nuclear and the mitochondrial DNA. They are the main topics of discussion, the nuclear and the mitochondrial DNA. Okay, let's quickly talk about the cell-free DNA and then nuclear. So if you try to imagine or visualize, this should be easy to understand what I mean by cell-free DNA. See, when you measure the enzymes in the blood, there are a lot of enzymes in the blood, but I told you large amount of those enzymes are actually not having any function. We call them the non-functional enzymes. However, they are present in the blood due to, what is the cause? Due to cell turnover. When the cells die, some of their content will spill into the blood and we can measure that blood and measure that enzymes, and we have a basal level of those enzymes based on the basal rate of cellular turnover. Now enzymes can come in the blood from the cell. Obviously the DNA can also come into the blood by the same mechanism. So essentially it is a mix of the cellular and the nuclear and the mitochondrial DNA which is coming inside the blood due to the cell turnover. When the cells die, some amount of DNA in the broken form will be available in the blood. Alright, so the DNA in blood is called the cell-free DNA. Now, what is the important new techniques which make use of the cell-free DNA? First, there is something called liquid biopsy. Now, whenever we are suspecting any malignancy in a growth, sometimes what happens in the body, there is an abnormal growth. The growth may be benign. Benign means it is growing very slowly and it is not going to create a problem. When it becomes big in size, we can just remove it. Or it can be malignant. Malignant means it has a cancerous potential. It can spread in the body and it can quickly start causing damage to the body because it uses up a lot of energy, and the other part of the body will be deprived of that energy. So that is the malignant growth. Okay, so whenever there is a growth, to distinguish between the benign and the malignant nature, what we do, we take a small biopsy and then we stain that tissue and we examine it under the microscope to determine whether the regular cells are there or there is a loss of the regular feature, which we'll discuss next year in pathology. So whether there is a pathological pattern of the cellular arrangement or there is a regular pattern of the cellular arrangement, which will determine whether whether the growth is benign or malignant. So that is the regular biopsy. We take the biopsy from the affected tissue. Now, you have discussed in anatomy the thorax, the neck part you have discussed, and there are certain regions in the body which are not easy to access or very risky to access. Suppose there is a growth, deep-seated growth in the lateral side of the neck. Now, is it advisable to approach that tissue to take a biopsy, only biopsy, just for diagnosis, even though it might turn out to be benign? No, it is very, very risky. You can see large amount of blood vessels and the nerves are passing through this, very critical nerves and vessels are passing. So it is very risky to take a biopsy from a deep-seated growth from the side of the neck. Similarly, if there is a growth in the retrosternal space, in the front we have the large vessels, in the back we have the intervertebral disc and the spine. So how to approach that interstitial space? It is very, very risky. You might damage any of these organs depending on which side you are approaching from. So again, we would not like to take a biopsy. That is where the liquid biopsy comes into play, with the understanding that whatever DNA will be there in that particular malignant cell will also be coming in the blood, because the malignant cells will also show a turnover. Some old cells will die, new malignant cells keep dividing rapidly, and because of that, the malignant cellular DNA will also be there in the blood. Depending on the site where the growth is there, we also can have an idea what type of the malignancies can be there, and later on we can have the specific test for that type of malignancy. So basically, liquid biopsy uses cell-free DNA for checking for malignancy. In very simple words, it uses the cell-free DNA for checking malignancy. Alright, that is one. Second thing, the cell-free DNA is also used for something known as non-invasive prenatal testing, commonly known as NET or NIET. Here the premise is the cell-free DNA in mother is 90% maternal and 10% of fetal origin. Alright, this is the premise for the non-invasive prenatal testing. So what we do, first we separate this 10% fetal DNA and then we examine this 10% fetal DNA for trisomies. We examine the cell-free DNA present in the mother's blood, which is coming from the fetus, for trisomy in the fetus. Otherwise, how do you determine it? You have to do the karyotyping. For karyotyping, you have to take the sample either from the by amniocentesis, you take it from the amniotic fluid, or by the chorionic villus sampling, we take a sample from the placenta, the chorion, and then we do the karyotyping. Both of them have their own risk. There can be loss of pregnancy. This is very safe because here we are taking the sample from the mother's blood. From the vein, you take a small blood sample and you separate the fetal DNA and you do the test. Okay, so these are two new techniques. Remember these names, sometimes they can land up in your viva. Can you tell me something about cell-free DNA, the examiner can ask, or sometimes in the clinical part. Clinical part again, these questions can be there, or just write a short note on liquid biopsy or write a short note on the technique, the NPT. So you should know there we are talking about the cell-free DNA. Okay, so let's go back to the nuclear and the mitochondrial DNA, which are the primary points of discussion in our discussion of the nucleic acid. Okay, so first let's talk about the nuclear DNA. The DNA present in the nucleus in the humans. The nuclear DNA. This nuclear DNA can be of different conformations. Conformation means different shapes can be there in this nuclear DNA. The most common conformation for nuclear DNA is the B form of DNA. So we'll start off with our discussion on the B form of DNA, and then we'll see what other types of DNA can be there. Some very frequently get the comparison between B form versus the second most common, that is the Z form of DNA. So let us first discuss the B form DNA and then we'll compare with the second most common form, that is the Z form of DNA. So when you get the question on write a short note on the B form of DNA. So there are certain points about the B form DNA that you should know. First point, we said it is the most common. It is the most common conformation of the nuclear DNA. It is the most common physiological conformation of the nuclear DNA. Now, what are the properties of the B form of DNA? First and foremost, it is double stranded. Alright, it is double stranded, and we have already said the two strands are anti-parallel. We had seen when we were drawing the double stranded DNA about the nucleotides, we had said the orientation of the two strands will be anti-parallel. They will run in 5'3' direction in opposite direction whenever we make the double stranded DNA. Alright, so the nuclear DNA is double stranded and the two strands are anti-parallel. The nuclear DNA is linear. It is important to mention the linear nature. Why? Because the mitochondrial DNA will be circular. See, the mitochondrial DNA is circular. Alright, and each of the strands, we said it is double stranded. So when you examine each of the strands have a structure like a helix. So we call it the double helical structure. Two helix running around each other, so it has the double helical structure. Now whenever you have the helix, alright, the helix can have two types of orientation. How it is running in a spiral, it can either run clockwise or it can run anti-clockwise. So the orientation of the strands of DNA are clockwise. When it is clockwise, we call it the right-handed helix. So the two strands of the BDNA are arranged in a right-handed manner, and when you examine the structure, you find the presence of grooves. This is very important. We find the presence of grooves. They are major and minor grooves. We find the presence of major and the minor grooves. So double stranded, anti-parallel, linear, double helical structure, right-handed, and the presence of grooves, the major and the minor grooves. I'll draw a representative sample. The representative sample, right? Representative sample of the BDNA. Representative sample of the BDNA. Here you have to make note of a few things. when you examine this structure, you have to make note of few things. First is the grooves. Minor groove, and then you have this major groove. Minor groove and the major groove. Right? First point. Second, what is the width of the helix? What is the width of the helix? Okay, this second point which you should know. Width of the helix, which is 20 Armstrong or 2 nanometer. The width of the helix is 20 Armstrong or 2 nanometer. And the third thing is, what is the distance occupied by one turn of this helical structure? Distance occupied by one turn of helical structure. Now this is where some of the books will differ in the information. Most of the books will mention 34 Armstrong containing 10 base pair per turn, but this is an approximation to simplify it for the UG level. Please note, although your MBS books will mention the 34 Armstrong or 10 base pair per turn, the more correct answer should be 36 Armstrong having approximately 10.5 base pair per turn. Now it is somewhat difficult to explain how come you have 10.5 base pair, but please understand what is happening here. The DNA is not an isolated, it is not having isolated curve, it is a continuously folded structure. So it need not have the exact multiples of nucleotides per turn. Alright, so it actually has 10.5 base pair per turn, and the approximate distance occupied is 36 Armstrong. Okay, so that distance occupied by one turn of DNA is called the pitch of DNA. So what is the pitch of BDNA? The pitch of BDNA is more correctly 36 Armstrongs or 10.5 base pair per turn, but your books will mention 34 Armstrongs or 10 base pair per turn. So pitch of the DNA is 34 Armstrong or 36 Armstrong depending on which source you are using. So this is the points about the B form DNA. It is double stranded, it is anti-parallel, the structure is linear, and it shows the arrangement of the double helix, and the helices are right-handed. And finally we find the grooves, the major and the minor grooves. The major and the minor grooves. The grooves you can see here, the major and the minor grooves. Okay, so some additional viva questions which are commonly asked here. What is the importance of the grooves? Why the major and minor grooves is an important property? Why are we highlighting the major and the minor grooves? See, because of the major groove, you can see the space here. The space here is much greater than half the width. Alright, the half the width would be let's say 10 Armstrong, but here you can see the distance definitely more than 10 Armstrong. The benefit of this major groove is the proteins which interact with the DNA. The proteins which interact with the DNA, even though they are bigger than 10 Armstrong, they can enter into the major groove and still interact with the DNA without disrupting the helical structure. So this is the main importance of the major groove. It allows for regulation of the replication, transcription without affecting the helical structure. We don't need to disrupt the helical structure. Because of the major grooves, the proteins can slide inside and they can still interact with the DNA. So this is the main point about the grooves. It allows for easy interaction with proteins. You know that there are several DNA binding proteins which have the DNA binding domain. So these domains are easily able to move into the major group and interact with the DNA. Alright. So this is about the B DNA. Now let us quickly compare it with the Z DNA in the same screen. How to compare with the Z DNA? Some of the points are similar. Some of the points are similar. The Z DNA is also double stranded. The Z DNA, the two strands are also anti-parallel, and it is also linear. And this is where the similarities end. This is where the similarities end, because the structure is not a double helical. It is having the zigzag shape. I'm sure all of you have read this in your 11th and 12th. The ZDNA has the zigzag shape. It has the zigzag shape. Alright, it has the zigzag shape, and its arrangement is not right-handed. It is rather left-handed. Means the strands run in an anti-clockwise manner. They run in an anti-clockwise manner, and they don't actually have grooves. We don't find grooves. We would like to look at the structure, the simplified structure of the Z form DNA. The arrangement of the nitrogenous bases is something like this. The arrangement of the nitrogenous bases is something like this, and this is from where we get the zigzag name. Alright, zigzag. You can see the arrangement is zigzag, and this Z is used to give the name of the Z DNA, zigzag. Within this distance, we actually have 12 base pairs. In the Z form DNA, we have 12 base pairs, and you can see there are no major and minor groups. One end is exactly coming to the end, whereas the other one is right up till the middle. Right, it's running right through the middle, so we don't have the major groups. So easy interaction with protein does not occur. The Z DNA is not amenable to good regulation. It is not amenable to good regulation. So very frequently we find in the exam that compare between the BDNA and the ZDNA. So these are the points on which you are going to compare and contrast. So what are the similarities and what are the differences between the BDNA and the ZDNA? What are the similarities and what are the differences between the BDNA and ZDNA? Alright. Now, sometimes you can get a general question, a 5-mark question, write a detailed description of the nuclear DNA. So, obviously, you will write about the B form DNA, you will write about the Z form DNA. At the same time, you should not forget that besides the B and the Z form DNA, there are some other conformations of the nuclear DNA, the A form, the C, D, or E form. So these are some additional conformations of the nuclear DNA. However, they are very similar to the B form DNA. Their basic properties are similar to the B form DNA. Only difference is in dimensions. Dimensions means the pitch of the different conformations will be variable. Otherwise, they will double stranded, anti-parallel, linear, double helical structure, right-handed, and will have the major and minor groups. Only difference is there in the dimensions for the A, C, D, and E forms. The A type will have 11 base pair per turn. The C, D, and E forms will have 8 base pair per turn. Okay. Otherwise, they will be very similar to the B form. You don't need to go into details for this. You can just mention that the other conformations which can be seen are A, C, D, and E. They have a short half-life, and properties are similar to the B form of DNA. Alright. Okay, so this is the broad general conformation of the DNA. But when you examine the DNA, we come across a very important problem, not only for the human nuclear DNA, but for any DNA, any organism. When you examine the DNA, we come across a very big problem. What is the problem? I'll take the example of humans only. See, in our body, let's take the example of average student. Typically, if you take a young first year student, male student, maybe the height of an athletic boy, a very athletic boy, can be at most 6 feet, or maybe 6.1 feet, 6.2 feet. So let's say 1.8 meters, right? Now, in our body, we know there are trillions of cells, right? We have trillions of cells. So, what I'm going to do, I'm going to take one single cell from this person. Take one single cell from this person, and I'll magnify this cell. Okay, I'm going to magnify this cell. When you examine it under the microscope, it appears something like this. Alright, I've magnified the view. Now, this cell, if you check the width of this cell, depending on which cell is this, whether it is the neuron or it is the RBC or which cell is this, the dimension can vary from 5 to 60 microns, and I hope you know that micron means the dimension of this cell. The diameter of this cell can vary from 5 to 60 microns, depending on which cell are we talking about. You have seen this in your physiology and your histology sections. The size varies from 5 to 60 microns. Now, within this cell, we have the nucleus, which is obviously occupying a smaller space in comparison to the cell. It is within the cell, so it will obviously have the smaller space. And within this nucleus, in one of the corners, all the different 46 DNA chromosomes are sitting. All the 46 DNA are sitting. Now, if you take out all this DNA, if you take out all this DNA, straighten it out, and put it one after the other. If you put out all the DNA one after the other, and like this you arrange all the 46 DNA particles one after the other, end touching to end, from one single cell. See, from the trillions of cells you've taken one single cell, and you measure the length of the DNA, all the DNA within one cell. What do you think would be the dimension? What would be the dimension of this particular structure? All the DNA particles arranged one after the other, end to end. 2 meters. The 46 DNA particles within the human nucleus, if arranged end to end, is measuring 2 meters. See, it is even longer than the person. And if you compare with the cell size, you find a great disparity in the total size in comparison to the place where it is stored. Another problem is you cannot just wrap it up and throw it in the corner. No, why? Because you need to access this DNA again and again. Every time you want the protein, you have to localize the gene on the DNA. You separate the two strands there, you carry out the transcription, then you do the translation, and only then you get the protein. The problem is more difficult when you want to make a new cell. When you want to divide the cells, the whole of the DNA strands have to be separated from one end to another for the replication to occur. If there is a knot at some place, game over. Separation will not occur, the replication will not occur. So you cannot just crumple up all this DNA and throw it one corner. There must be a very proper sequential and orderly arrangement of the DNA so that it can be compacted and stored in such a small place that we have described. Alright, so what we're going to look at now is the arrangement of the DNA inside the nucleus. How the DNA is arranged inside the nucleus in the eukaryotes. Particularly, we are talking about the humans, but it is applicable for most of the eukaryotes. In prokaryotes, because the nucleus structure is not there, such well-defined organization may not be seen. Still, some arrangement of the DNA does occur in the prokaryotes. However, we are focusing on the arrangement as seen in the eukaryotes. Okay, so what happens when you look at the arrangement? When you look at the arrangement of the DNA in the nucleus, we find that the compaction in size occurs multiple times, right? So we talk about the level of compaction or the level of arrangement, and we find that there are multiple levels of this arrangement, multiple levels of this arrangement, starting from the double helical DNA structure which is there, how the different levels are organized, and they are compared to the geometrical shapes or structures or natural shapes and structures to give us a bird's eye view how the organization is occurring. So I'll briefly mention how the organization is occurring and then go into the details of the first and the last levels. Okay, very briefly I'll mention the organization at all the levels and then go into the details of first and the last levels. The first level of organization resembles a structure called solenoid. I am sure all of you have read about this structure in your physics in electricity. What is solenoid? Solenoid is a structure where we have a central cylindrical core around which the wire is wrapped. Right? So don't worry, we'll go into the details of this structure very soon. We'll go into the details of this structure very soon. So the first arrangement of the DNA is in the form of the solenoid. Alright. So first arrangement of the DNA is in the form of the solenoid. The solenoid arrangement, when you look at under the microscope, appears something like this. This is the solenoid, and intervening segment is there. Then again the solenoid is there, the intervening segment, the solenoid, intervening segment. Like this, the arrangement is there. The intervening sequences of DNA which are not the solenoid, and then the repeating solenoidal structure is there. These solenoidal structures rearrange as a solenoid. So the solenoid arranging as solenoid, we call it the super solenoid. So what happens? They arrange again as solenoid. However, this time there is nothing in the core. What happens? The nucleosomal structures, sorry, these solenoidal structures start getting compacted near each other. Alright. In each turn, there is approximately seven solenoidal shapes which are compacting themselves together, and they form this super solenoid structure that I have drawn. Right. So next is the super solenoid structure. Now think of this super solenoid structure as a thread. If you extend it very far, it will become a thread, although it will be a thick thread, but it can still behave as a thread. And this super solenoid will then arrange as loops. The thread will form loops. So now the structure is something like this. Each of these thread-like structure will form the loop. So the loops are formed. The loops come together to form walls. Flower-like structure, you see flower, there is central body on which the various petals are there. We call it the wall of flower. So something like that. the whorls are formed with the help of the loops. Alright, so we call it a whorl. The whorls are stabilized by certain structures. We call these structures as nuclear scaffold. We call the structure as nuclear scaffold. Right? And multiple whorls will arrange one after the other. Behind this, you will have another whorl-like structure. See something like this. Right. So multiple whorls arranged one after the other reform a solenoid, and then the super solenoid. Now, this further arrangement as solenoid and super solenoid may or may not be seen sometimes. The arrangement may end with the whorls only, or it can further be organized as the form of the solenoid and the super solenoid. The further compaction is there if we go to the different stages. Okay, so very commonly we find the 6 levels of organization. Every time there is a compaction, the amount of DNA present in a small given area will increase manifold. Right. So this is the organization of the DNA very briefly. I told you we'll talk in detail about two of them. The solenoidal structure that is the first level of organization has its own name. It is known as the nucleosome. And the last level of organization, whether it is the whorl, whether it is solenoid or the super solenoid, it is known as the chromatid. The intervening stages have not been given name, they just are compared with the structures that I have mentioned, but the first and the last level of organization have their own names. The first and the last level of organization have their own names. They are called the nucleosome and the chromatin respectively. The nucleosome and the chromatin respectively. And we'll talk about the nucleosome and the chromatin. Just 1 second, I'll switch on the charger and come back. Okay. Okay. So the nucleosome and the chromatin are the two structures that we are going to be talking about. Now, let's take them up one by one, starting with the nucleosome. Okay, so nucleosome, like I said, is the first level of organization of the DNA. Nucleosome is the first level of organization of the DNA, and we already said it has the arrangement similar to a solenoid. It has the arrangement similar to a solenoid. In the solenoid, we've already seen there is a central core. There is a central core around which the DNA is arranged. Around which the DNA is arranged. Alright, this central core that you see is made up of histones. More precisely, the structure is a histone octamer. The structure is a histone octamer. The name itself tells you there are 8 subunits of histone. Alright, there are 8 subunits of histone. It's called the histone octamer. Now, if you examine the histone octamer, what will you find? H2A attached to H2B. H3 and H4, that makes 4. We need 4 more. There should be 8. So they are present in duplicates. 2 copies. The dimer the dimer. Interestingly, sometimes we can have dimer of H3 and H4 also. Alright, we can have the dimer of H3 and H4 also. Together, this is the histone octamer. Together, this is the histone octamer. We'll talk about histones very soon, don't worry. We'll come back to the topic of histones. So we have the histone octamer, around which the DNA is wrapped. Around which the DNA is wrapped. If we examine the wrapping, we will find 1.75 turns. It is not a complete 2 turn. It is not a complete 2 turn. It is 1.75 turns, containing 146 base pairs. The loop of DNA around the histone octamer is occupying 1.75 turns and contains approximately 146 base pairs. It contains approximately 146 base pairs. Obviously, DNA is a long structure. It is not made up of only 146 base pairs. So, this nucleosomal structure cannot be alone. There must be other nucleosomal structures, obviously. So, we have another nucleosomal structure here nearby. And obviously they will be linked. So this is a second nucleosomal structure and they are linked. It is a continuation. It is a continuation of the DNA. It is a continuation of the DNA. This DNA, which is intervening in between the two nucleosomes, is known as the linker DNA. This is known as the linker DNA. The linker DNA is not exposed. Please note, the linker DNA is not exposed. It is also associated with a type of histone, which is the H1 histone. This is the H1 histone. So linker DNA protected by H1 histone is lying in between the nucleosomes. It is lying in between the nucleosomes. We can have another nucleosome here. We can have another nucleosome here. Right? And the DNA, which is wrapped around the histone and the linker DNA, protected by the H1 histone. Protected by the H1 histone. Right. So this is the structure that we see. This detailed structure obviously not visible under the light microscope. Multiple reasons. First, we had said the nucleotides absorb the UV light, so they are not visible to the naked eye. Number two, the size is very small. It is running in the nanometer and Armstrong, so obviously it will not be visible to the naked eye. Anything which is smaller than microns, it is not readily visible to the naked eye. So not possible to see this detailed structure under the light microscope. So what do we use? We use the contrast type of electron microscope. We use the contrast type of electron microscope. You know, the contrast type of electron microscope is giving us the computer generated images based on the intensity of the color that we get. It will impart colors to the various particles so that we can make out the detailed structure. So contrast type of electron microscopy, which gives us more detailing, is only able to decipher so much detail. If you don't use the contrast type electron microscope, rather if you use the regular electron microscope where we don't have the reconstruction of images, in this case what you see, what I have shown previously, the nucleosome, linker DNA, 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 something like this. This is called beads on string. Under the regular electron microscope, we get the beads on string appearance for the nucleosomal structure. Please note, even the beads on string appearance cannot be seen by the light microscope. We will still need the electron microscope, the regular type of electron microscope. Okay, so beads on string appearance under the regular electron microscope, and in the contrast electron microscope, we see the solenoidal shape, the solenoidal arrangement of the nucleosomal structure. Alright, the linker DNA is said to contain approximately 54 base pairs. It is said to contain approximately 54 base pairs. Now here I'll tell you a very important concept regarding the cellular turnover, which can be asked sometimes from you in the viva, and it will definitely be useful for you next year when you go to the pathology section. So see what happens when the cell damage or cell death occurs. It can occur in two ways. There can be planned cell death, we call it apoptosis. There can be unplanned cell death, we call it the necrosis. So we have apoptosis and necrosis, the planned and the unplanned cell death. So when there is planned cell death, there is break in DNA at specific locations. There is break in DNA at specific locations, whereas where there is necrosis, break in DNA is random. It will occur at any and every positions. When we say specific location in apoptosis, we find that these locations are present within the linker DNA only. The DNA is not cleaved over the nucleosome, rather it is cleaved only at the position of linker DNA. So what happens? Suppose one cut is made here and the another cut is made here. So we'll get a fragment which has approximately 4 nucleosomes. In another segment there can be another cut over here, let's say. So we can have a fragment of DNA having 3 nucleosomal length. If we get a cut here, we can get a segment of DNA having 2 nucleosomal length, and we can also have the segment of DNA having 1 nucleosomal length. So when we examine the pattern of DNA in the gel electrophoresis after apoptosis, we get a very specific peculiar pattern. We get a very specific peculiar pattern. From the point of application, we find regular multiples of size. We find the regular multiples of size. We find the distance between each fragment keeps decreasing as we go close to the top. Right. So this is the ladder pattern. We call this the ladder pattern of electrophoresis, which is peculiar for apoptosis. Whereas for necrosis, the damage can occur anywhere. It can cleave here also, it can cleave here also, it can cleave here also, it can cause the cleavage anywhere. So in that case, what happens? When we examine the pattern of arrangement, we find fragments of all sizes. We find fragments of all sizes. This is the smear pattern. Smear pattern on gel electrophoresis is characteristic for necrosis. If the cell death is due to necrosis, when you do the gel electrophoresis for the DNA, you will find the smear pattern. If the cause of cell death is apoptosis, when you do the electrophoresis for the DNA, you will find the ladder pattern. Alright, so ladder pattern and smear pattern of the cell fragments after cell death. So in apoptosis, it is multiples of nucleosomes. So the farthest one, maybe it has 1 nucleosome, this one has 2, this one has 3, 4, 5, 6. So there will be the multiples of the nucleosomal size. So if you look at the sizes that we had mentioned previously, 146 plus 54. So one nucleosomal fragment is approximately 200 base pair in size. One nucleosomal fragment is approximately 200 base pair in size, and that is how we get the multiples. The multiples are approximately 200 base pair in size in the apoprotic ladder pattern. Okay, so this is about the nucleosomal arrangement. I told you I will talk a little bit about the histones. What are these histone particles around which the DNA is arranged? For this you have to understand the nature of DNA. If you recall, the nucleotides have large amount of phosphate. The phosphate is forming a bridge between the address bases. The backbone of the nucleic acid is formed by phosphate. What is the charge on phosphate? Negatively charged. In other words, it is acidic. So because of the presence of phosphate, the DNA is acidic in nature. Not only DNA, RNA is acidic in nature. And if it is going to make a very close attachments with any protein, it must have the opposite nature. So DNA in nature is acidic, therefore the nature of protein with which it attaches must be basic, and that is why we find that the histones are basic proteins. The histones are basic proteins. We already said when you're doing the amino acids that there are 3 amino acids which can be basic in nature. Out of them, 2 amino acids are present in very large amount in histone. They are lysine and arginine. The basic nature of histones is due to the presence of large amount of the lysine and arginine. Please note that histidine is not present in significant amounts within the histones. Histidine is not present in significant amount within the histones. Mainly it is the lysine and arginine which is responsible for the basic nature of the histone proteins. We already mentioned the different types of histones: H1, H2A, H2B, H3 and H4. Alright. Some extra point about histone which you should know: there are no introns in genes for histones. This is the unique eukaryotic protein where the gene doesn't contain the introns. Almost all the genes for all the proteins will contain introns except for the genes for the histones. Now, within the nucleosomal structure, within the DNA, the histones show a very important property which is responsible for the optimum function of the DNA, and that is the covalent modification. The histone proteins readily undergo covalent modification under optimum conditions. Under optimum conditions, they will show the different type of the covalent modification. What are the different types of covalent modification that you may see? Most well studied covalent modification is the acetylation. Find the acetylation. We find phosphorylation. We find the ADP ribosylation. ADP ribose is added. This is called ADP ribosylation. We find the methylation. We find attachment of single ubiquitin. It is called monoubiquitination or quitination. Both terms are used. Monoubiquitination. Then there is attachment of proteins called sumo. I'll tell you what are those proteins. And the phenomena is called sumoylation. Sometimes citrulline can be added, and the phenomena is called citrulination. Here there is attachment of SUMO. SUMO stands for small ubiquitin related modifiers. SUMO stands for small ubiquitin related modifiers. They are ubiquitin like proteins, but they are smaller in nature, and we call them SUMO. When the SUMO is attached, the phenomena is known as the SUMOylation. They are the covalent modification, means they get attached covalently with the histone particles. What is the importance of this covalent modification of the histones? Before I go into the detail and the importance of covalent modification, please note that there is also the covalent modification, thank you, of DNA, and we see only one type of covalent modification of DNA, and that is methylation. In the DNA, only type of modification that you see is the methylation, demethylation. Other covalent modifications do not occur in the DNA. Now, what is the importance of these covalent modifications? Why I'm telling you about these covalent modifications? See, when you want to carry out the transcription or even translation, you have to remember that the DNA is not present in the helical form in the relaxed state. Why? We have already seen that it is wound around the histone proteins because of their opposite charge. They are tightly linked to each other. Sorry. Because of their opposite nature, they are very tightly attached to each other. As long as they are attached to the histone, you cannot separate. You have to first separate the strands of DNA. Only then you can carry out the transcription replication. So as long as they are attached to the histone, you cannot carry out the separation. So first they have to be dissociated from histone. Only then the two strands can be separated. Alright. So separation from the histone will depend on what is the degree of attraction. If the segment of histone where the DNA is attached is very basic, then the attachment will be very strong. If the degree of basic nature is less, then the attachment will be weaker and separation can occur more easily. In other words, the replication transcription can occur more easily. Alright. Now imagine there is a protein which is basic in nature, and now you add the acidic group phosphate on it. What will happen to its basic nature? Obviously the basic nature will decrease. When the basic nature will decrease, the attraction between that segment of stone and the DNA will decrease. Therefore the separation of that segment will be easier, and the transcription, even replication in that segment can occur more easily. We are more concerned with the transcription part, not the replication part. So transcription can occur more easily in that segment. In other words, the genes present in that segment will be transcribed more frequently. Alright. So what we are doing here, the covalent modification of histone and DNA. The covalent modification of histone and DNA is able to regulate the process of transcription. If the transcription is regulated, automatically the translation will also get regulated. Alright. So it is resulting in a regulation of transcription. Now this is very interesting and a different information from what we traditionally know. What we traditionally know, all the information related to the DNA, RNA and protein is contained in the sequence of the nucleotides. We call it the genetic information. Alright, we call it the genetic information. The information contained in the sequence of nucleotides is the genetic information. But here, the covalent modification which is able to regulate the transcription, it is not a function of the sequence of nucleotides. It is a function of covalent modification of the histones and to some degree the DNA also, without changing the sequence of nucleotides. Nowhere is the sequence of nucleotide coming into the picture. What we are talking about is the covalent modification of the various segments, whether it is the DNA or the protein. So this information which is being conveyed is not contained in the genetics. It is extra information. It is beyond the genetics. It is over and above the genetics, and that is why the term used is epigenetics. The information conveyed by covalent modification of histones and DNA is called the epigenetics. And a very important point to understand is this covalent modification is easily regulated by environmental factors. It is easily regulated by environmental factors, availability of nutrients, whether it is hot, whether it is cold, whether it is acidic, whether sufficient amount of hydration is there or not. So it is regulated by environmental factors. The covalent modification is regulated by environmental factors. So what happens? We have the genes, the sequence of nucleotides, right? We have this genes, the sequence of nucleotides, but their transcription to some extent is being regulated by epigenetics. Only when the transcription is completed, and depending on at what rate the transcription is completed, we'll see the final expression, that is the phenotype. Alright, and epigenetics is affected by the environment. Alright, so epigenetics is called the gene environment bridge. It is called the gene environment bridge. Try to understand it in this manner. There are a lot of diseases where we say that multiple genes are involved in causation of disease. That is why we cannot predict easily that at what time the disease will happen. Alright, so we have the polygenic theory of disease. Most of the non-communicable diseases are said to occur due to multiple genes. Multiple genes are involved and therefore they cause the disease. Now, suppose we have two specimens where the genetic sequence is exactly the same. We have that in nature also. We have the monozygotic twins. In the monozygotic twins, the genetic sequence is exactly the same. So even if we are talking about a polygenic disease, multiple genes are involved in the causation of disease, when we have the two specimens where exact the same genes are there, then a disease which is affected by multiple genes should occur simultaneously because the genes are same. Meaning, if you have a pair of monozygotic twins, as per the polygenic theory of disease, if you have a pair of monozygotic twins, then twin A on, let's say, 24/7/2020 has been diagnosed with type 2 diabetes mellitus, which is a polygenic non-communicable disease. So as per the polygenic theory of disease, on the same day, the second twin should also have the type 2 diabetes mellitus. Same genes are there, but it doesn't happen. It doesn't happen. Why? Because the environmental interaction, their lifestyle, how they have interacted during the time till they have come to maybe 45 years of age, 50 years of age, their environmental interaction is different, and therefore the second twin may not have the disease, or he might have already developed the disease. He will not develop the disease on the same day because they are having the different environmental interaction. So this environmental interaction is regulating the expression. That is why the epigenetics is now a very, very important topic, which tells us it is not the genetics which will decide the final phenotype. It is also the conditions in which this genotype is getting transcribed, which is under the regulation by environmental factors, and that regulatory switch is the epigenetics. So please understand, epigenetics is the modification of histone and DNA which is able to regulate the transcription of the genes, thereby affecting the final phenotype. So epigenetics is a very important topic. In the last 5-6 years, we have seen in most of the universities very frequently the question has come on epigenetics. Alright. So you should know about the epigenetics, and you should remember the different types of covalent modification that are seen inside the histones, and that too in the DNA. In DNA, we said only the methylation will be seen. Only the methylation will be seen in the DNA. This methyl group can be removed, it can be added. Methylation, demethylation. But for the histones, we have 7 different types of modification that can be seen. Now, the modification may increase the rate of transcription, or it may decrease the rate of transcription. It is not fixed. It depends on which DNA we are talking about, whether it is deep inside or it is on the outside, which type of histone is there. So various factors affect the net result of covalent modification on the histone. But methylation always decreases the rate of transcription. Methylation of DNA always decreases the rate of transcription. The covalent modification of histones has variable effect depending on the type of histone, position of histone, and in which chromosome this change is occurring. Okay, so this is about the epigenetics. Epigenetics, I said, is a very important topic, which is based on the histone part of the DNA. It is based on the histone part of the DNA. And this completes the discussion of the first level of organization, which is the solenoid. Okay, solenoid, which is the nucleosomal structure. We have seen the nucleosomal structure has the cylindrical part, which is the histone octamer, and the thread part, which is 1.75 loops of DNA. Various nucleosomes are linked together by the linker DNA, having 24 base pairs surrounded by the H1 histone. The detailed structure is visualized under the contrast electron microscope. If we use the regular electron microscope, what we get is the beads on string appearance. Alright, the various histones present in the histone octamer are H2A, H2B, H3 and H4. Two copies each of them are present. Histones can undergo modification, the covalent modification, along with the methylation of DNA. Together, they result in epigenetics. They are able to regulate the rate of transcription, and we call it epigenetics. Epigenetics is the gene-environment bridge. This is how the environmental stimulus will regulate the genetic expression to produce the final phenotype. Alright, so that's about the nucleosome. Let's take a short break here, and after the break, I'll talk about the chromatin structure, and then we'll move on to the RNA. Alright, okay. Short break, and then we talk about the chromatin structure. After the chromatin, we'll talk about the RNA. Oh no, we also talk about the mitochondrial DNA. We also have the mitochondrial. Alright, so please come back after 15